Hello, my name is James R. Mickelson. I was part of the research team from the University of Minnesota in the 2013 publishing of PLOS Genetics. I run the Canine and Equine Genetics Lab at the university. For this publishing, I examined 33 breeds of horses, including quarter horses and paint horses. Through examining these breeds, we were able to link the genome for myostatin, the speed gene, to the genome of the quarter horse and paint horse through their engineered matings. The myostatin gene contributes to muscle mass, which does play a role in speed. Though this be a contributor to the speed gene, there are many other factors that contribute to performance in horses. These include oxygen absorption of the muscles, thickness and morphology of the bones, and their heart size. For part of the experiment, we collected muscle biopsies from horses and examined them for two different types, slow twitch and fast twitch. Fast twitch contracts more rapidly and with greater force. Slow twitch is less rapid and can convert glucose to fatty acids more efficiently. The fast twitch and slow twitch muscle types determine a horse's predisposition for sprint or endurance sports. Through this testing, we came to the conclusion that the speed gene does not cause a shift in fiber types and does not change the size of muscle fibers. However, we do know that the speed gene is known to play a role in a horse's athletic performance.
This is Jim Bolger, a legendary horse trainer. In June of 2016, his horse Trading Leather won the Irish Derby. He knew Trading Leather would win because he had him tested for the speed gene. Now Jim Bolger promotes the speed gene testing for everyone that he knows.
Hello, my name is Emmeline Hill. I'm a researcher at UCD. Equine genomics is my area of study. To start learning about the speed gene, I started by collecting DNA samples from multiple thoroughbreds from horse farms in Ireland. I then found a gene with a single genetic determinant for speed. I then proceeded to look at the gene myostatin, which inhibits growth of muscle fibers by limiting proliferation of muscle cells.
Emmeline Hill is one of the scientists/geneticists that helps in the finding of the speed gene in the horse. At a young age, she had an interest in horses due to her grandma, who was the very first woman to race against men in Ireland. And since then, she had many opportunities to help grow her passion in genetics, and especially kickstarting the speed gene she thought about in horses would be the myostatin in mice, and looking at that and asking the same question for horses. The amount of myostatin, could it affect the racing ability? And so from there, she was interested in seeing if genetic types were more common in winning horses, such as your thymine, adenine, cytosine, guanine, but she wanted to know between double cytosine, cytosine paired with a thymine, or double thymines. So she took samples of 142 racehorses and ran tests to see if their genetic type would correlate with the race length. Turned out there was a very strong correlation between the two. Her results showed that base pairs double CC were sprint racers from 1,000 to 1,600 meters, the CTs were best at 1,400 to 2,400 meters, and the TTs were best at 2,000 meters. Ms. Hill's research was published in 2010, and it astounded all scientists that a single gene could predict best races, and thus started the existence of the speed gene.
So we know what the speed gene is, but how does it work? So the speed gene is a myostatin deficiency. Myostatin over here, so this is a regular muscle fiber, and what myostatin does, it's in myoblasts, and myoblasts bind to the muscle fiber to inhibit the growth of muscle fibers by limiting the proliferation of muscle cell precursors, aka myoblasts. Since larger muscles require more calories to sustain them, some have suggested the protein would have conferred an evolutionary advantage by ensuring the size of the muscle remains appropriate and efficient to an animal's activity level. So when it comes to the speed gene and there's a lack of myostatin, this is a normal muscle with myostatin, but then without myostatin, this is not drawn to scale, obviously, but just a visual representation of how the growth can get a little out of hand, and since that's what they derived the speed gene from. Because normally, when a muscle happens, there is an action potential arrives. So normally, action potential arrives at a neuromuscular junction. So then, acetylcholine, which is this, is released but binds to the receptors and opens sodium channels, leading to an action potential in the sarcolemma, which is down here. This is the sarcolemma. And then action potential travels along the T-tubules. Hence, technical technical difficulty, that's fine. Then you have troponin up here, which the thick filament interaction leads to muscle contraction. So the muscle shortens and produces tension. That's what would happen in a normal muscle. But without the myostatin and the muscle being larger, more muscle equals more power, and that is the explanation of the speed gene.
Welcome back. So, on the speed gene, we've already talked about how it works and what it is, but we haven't talked about how it was tested. I mean, I guess you can call it testing, but it was more of like looking at certain genome lines and seeing what kind of matches up and what looks right. So, the scientists here decided that maybe it was something with like repeating STRs or something that stands out, and it looks like it could create the speed gene or the myostatin, whatever it is. So here we're showing an example of a buff horse and a limpy horse. And as you can see, the buff horse has a lot of multiple cytosines for their STRs, and that just shows that that will do the increase in the myostatin. As to over here, limpy horse doesn't have any of those, so he's not very muscular and he's not going to run very fast. But this guy, he's going to run pretty fast. It's like the Usain Bolt of horses. That's all.